Hello, my name is Dr. Roderick L. Roll, and today I will be talking to you about lipids. We are looking at our nutritional guide again. According to the science community, we are supposed to consume 28% of our calories in the form of lipids. It can be from animals, it can be from plants. Some of them are essential fatty acids, and some of them are cholesterols. And out of the total, we're supposed to have 65 grams of lipids per day. Lipids contain carbon, hydrogen, and oxygen. But the proportion of oxygen is less than it is with carbohydrates. These are some examples of lipids. We have neutral fats, which are also called triglycerides. We have phospholipids, and we have steroids. The first one we will talk about is triglycerides. They are also called neutral fats. This triglyceride will have a glycerol head and 3 fatty acid chains attached to the head through a modified covalent bond. This covalent bond will be called a ester linkage or a ester bond. If you look at the image below, we see a dehydration synthesis reaction that is occurring between the fatty acid tail and the glycerol head. The hydroxyl groups of both of these molecules get together and H2O is released. The tails of a triglyceride can be saturated or unsaturated. Saturated fatty acid tails contain all single covalent bonds, whereas the unsaturated fatty acid tail does not. In the unsaturated fatty acid tail, you will see a kink or a double covalent bond. Here is an image of a typical triglyceride. The first 2 tails are saturated. They contain all single covalent bonds, whereas the third one has one double covalent bond, or a kink. Therefore, it is unsaturated. We will be looking at 2 locations of these triglycerides. Animals contain one type of triglyceride, and plants contain a different type of triglyceride. The difference is based on the fatty acid tails. In animals, you'll tend to see more saturated covalent bonds, whereas in animals, you'll see more kinks or unsaturated bonds. In animals, the fats tend to have a high proportion of saturated fatty acids. This will make the animal fat appear solid. An example of a solid fat that was achieved from animals is lard. It is a solid lipid. Plant lipids tend to be low in saturated fatty acids. Therefore, they have more kinks. The kink, or the double covalent bond, adds distance between the 2 adjacent tails. This distance creates a more fluid-like material. So therefore, plant lipids tend to be liquid-based, or we call them oils. An example of one such lipid is corn oil. Converting unsaturated fats, which come from plants, into saturated fats require the addition of hydrogen. This sometimes creates what is called trans fats. This is that geometric isomer that we talked about earlier. So the process is called hydronation. So hydrogenated vegetable oils turn into things such as margarine. And in the image below, you see that the thing that appears to be butter is really a plant oil that has been hydrogenated. They added hydrogen. In the image to the right, you see the unsaturated fatty acid has kinks, so therefore it is in liquid form. But by adding 2 hydrogen bonds, you can straighten this kink up and it will appear like the saturated fat above it. But sometimes in the process of adding the hydrogen, you create a geometric isomer. This isomer is called a trans fat. For those of you that forgot the geometric isomer, here's the image again. If we hold one of the carbons in this image and rotate the other carbon 180 degrees, we change the spatial arrangement around that same covalent partner. This is a geometric isomer. So therefore, in the fat example, the tail would appear to be straight, therefore forming a solid, but it has the double kink still, or the double bond. Previously, we mentioned the negative effects of trans fats. So trans fats cause arteriosclerosis. It lowers the good cholesterol, and it overall increases cholesterol. Neutral fats are found up under our skin, so it is subcutaneous. It is also found around our organs. So in this image to the right, this is a cross section of skin, which is composed of the most superficial layer, is called the epidermis. Below that is the dermis, and deep down in the bottom where it's yellow, that is our hypodermis. In the hypodermis is where our neutral fat will be found. Another name for neutral fat is triglyceride. By utilizing this piece of equipment on the left, you can measure the thickness of your neutral fat. The second type of lipid we will talk about is the phospholipid. This phospholipid will be a modified triglyceride. Instead of having 3 fatty acid tails, it will have 2 fatty acid tails. In addition, it will also have a modification done to the head. This head will have a negative phosphorus group. This negative will make the head of this phospholipid polar. Therefore, it will be attracted to water. Remember, water has a slightly negative and a slightly positive portion. So water will be attracted to the head, but it will be repelled from the nonpolar tail. So again, the head is attracted to water and the tail is not. So another term you will see used in talking about phospholipids is hydrophobic and hydrophilic. The reason they use these terms is because the phospholipid has 2 sides. It is amphipathic. It has a side that is attracted to water and a side that is repelled from water. In a phospholipid bilayer, the phospholipids will orientate themselves in such a manner that all of the heads that are hydrophilic will face the water, and all of the tails, which are hydrophobic, will face one another. The third type of fat that we will discuss is steroids. The steroid structure is very unique. It consists of 4 fused lipid-based rings. The liver can convert a sugar intermediate called acetyl-CoA into cholesterols. In the image below, I show you the several step process to achieve the formation of cholesterol. In this image, I also show you the biochemical pathway to take glucose and convert it into cholesterol. So again, one of the intermediates in breaking down glucose is acetyl-CoA. Acetyl-CoA can be used to make cholesterol. Cholesterol is converted into bile salts, vitamin D, sex hormones, and adrenal cortical hormones. So you need cholesterol in your body. If you do not have cholesterol, then you cannot make these things, such as bile salts, vitamin D, and so on. So therefore, it is imperative that you have cholesterol. That's why the body has the ability to synthesize cholesterol if you're not consuming it. So again, we're going to discuss how cholesterol can be converted into these sex hormones called testosterone or estrogen. To discuss some of these molecules that cholesterol will be turned into, we need to learn a little anatomy and physiology. So deep on the back of a human, you have the kidneys. And on top of the kidneys, you have what is called the adrenal gland. In this adrenal gland, we're going to cut it open and look at the surface of it, which is called the cortex, versus the middle, the interior of that gland, which is called the medulla. So the cortex of the adrenal gland is going to produce about 24 hormones. These hormones are derivatives of cholesterol. This means that cholesterol is going to form all of these different hormones that we need in our day-to-day activities. The first portion of the cortex that we will discuss is the most superficial. The most superficial is the Zona glomerulosa. This is going to produce these mineralocorticoids. These mineralocorticoids are going to control mineral and water balance in the blood. So therefore, if you have an imbalance of mineral deposits or an imbalance of water in your blood, it could be a adrenal gland issue, or it could be you're not consuming enough cholesterol to adequately supply these mineralocorticoids. The zona fasciculata is the interior portion of the cortex. It will produce glucocorticoids. These will control metabolism. So individuals who have weight issues and they have problems controlling their metabolism, their catabolism of sugars and the anabolism of sugars, they might have a cholesterol issue. They might not be consuming enough cholesterol, which cannot make enough glucocorticoids. The most interior of the three is the zona reticularis. This will produce the Gonatocorticoids. These are involved in the development of puberty, sex drive in women, and estrogen production after menopause. So therefore, sometimes these individuals are having issues. It's because of the inadequate amount of cholesterol that is in their body. So therefore, they cannot make the appropriate gonatocorticoids. In this slide, we are trying to show you the importance of cholesterol. Through this flow chart or branch diagram, you see that cholesterol is going to make an intermediate called DHEA, which will then create testosterone. And if you are a female, you will take testosterone and convert it into estrogen. All of this was achieved through you having the appropriate concentration of cholesterol. These are some of the effects of the estrogen you just created from consuming the appropriate amount of cholesterol. Estrogen affects the brain, breast, bone, vagina, ovary, uterus, heart, and liver. So it is very vital for the survival of females to consume the right amount of cholesterol. Cholesterol is also very important for men. It is used in skin, brain, bone marrow, bone, muscle, and the development of the sex organ. So it's very vital for guys to consume cholesterol. So we all need to ingest cholesterol. Where can we get some of this cholesterol from? We can get it from eggs, meat, and cheeses. But we need to consume no more than 300 milligrams per day. Again, remember the structure of these cholesterols. These steroids will have these 4 interlocking rings. They're very different from any other macromolecule we have seen so far. This is the recommended daily intake. This was achieved or received directly from the Food and Drug Administration. You need to consume about 300 milligrams of cholesterol a day. We have a fat stain. This fat stain is called Sudan 4. This fat stain is a non-polar molecule that is red. It will stain other non-polar molecules red if you mix them together. If no fats are present in the solution, then this non-polar stain will aggregate, form clumps, and fall out of the solution and settle to the bottom. So in this image, we have several other fat stains also, Sudan 1, 3, 2, and 4. We will use Sudan 4 in lab to stain lipids. A positive stain will show the mixing of the nonpolar red stained molecule with your nonpolar solution. If your solution does not have a nonpolar molecule, then it will not stain. It will aggregate and form clumps and fall out of solution.